Hi everyone, in this USP series video, I am going to brief United States Pharmacopoeia Journal chapter 1220, Analytical Procedure Life Cycle. Traditionally, analytical validation was treated as one-time activity. Develop the method, validate it, and move on. That mindset is no longer sufficient. Today, regulators and pharmacopoeias expect us to think in terms of analytical procedure life cycle. This means the method is designed, qualified, monitored, and improved continuously over its entire life. The goal is not just compliance, the real goal is decision quality, ensuring that every reported result supports the right regulatory, manufacturing, and release decisions. Ultimately, this life cycle approach protects patient safety by reducing the risk of incorrect decisions caused by analytical error, variability, or method drift. So, we move from is method validated to is method consistently fit for the purpose. Analytical validation is no longer one-time checkbox. Today, the focus is on how an analytical procedure performs across its entire life, from design to qualification to routine use. This is a science and risk-based life cycle, not just a compliance exercise. The objective is simple: ensure reliable decision every time the method is used. Because when analytical data drive critical decisions, they ultimately impacts product quality and patient safety, no just regulatory approval. Life cycle thinking in USP 1220. It covers the entire method life, not just validation. Built on quality by design, risk-based, science-driven, this chapter introduces a life cycle framework aligned with ICH and FDA guidelines. It applies to all analytical methods from development to routine use. What is analytical procedure life cycle? It starts with the analytical target profile, that is ATP. Stage 1, procedure design. Stage 2, performance qualification. And stage 3, ongoing verification. The ATP guides every stage, and the stages are iterative, not rigid or one time. Validation is not a single event. It begins during method design, it continues through routine use, it includes validation, verification, and method transfer. Lifecycle validation replaces the old one-time mindset, shifting from a single study to continuous assurance. Analytical target profile ATP defines required method performance. It is linked to intended use. It focuses on quality of the reportable value. The ATP is defined before method development and it drives every design choice. ATP and decision making. The reportable value drives decision. Accuracy matters. Precision matters. The risk of wrong decision is explicitly considered. For quantitative methods, ATP defines how accurate and how precise results must be. So, regulatory and manufacturing decisions are trustworthy. ATP example: accuracy and precision can be defined separately or combined. As total analytical error, ATP should be technology independent. When possible, the goal is flexibility with the clarity, not complexity. Bias and precision. The core matrix. Bias is systematic error. Precision is random variability. Both directly affect the reportable value. Most performance characteristics trace back to these two, bias and precision, which together determine result reliability. Total analytical error. It combines bias and precision. It defines the allowable spread of measurements. An alternative approach is measurement uncertainty. Total analytical error captures the combined impact of systematic and random error on the reported results. Specifications and decision rules. Results are compressed to acceptance limits. Simple decision rules are most common. Risk increases as result approaches the limit. A decision rule defines how we judge compliance and how much risk of a wrong decision we are willing to accept. When do decisions become risky? Clear pass or clear fail, low risk. Results near the limit, higher risk. Variability drives uncertainty. When results sit close to specifications, analytical variability increases the chance of wrong decisions, leading to OOS results and even recalls. Guard bands and indecision zones. Guard bands reduce decision risk. Indecision zones trigger retesting. Decisions are controlled using a risk-based approach. By applying total analytical error, guard bands and multi-stage testing help manage risk advanced yet practical. Stage 1 Procedure Design. Select the right technology, understand the analyte and the metrics, design the method to meet the ATP. This stage is about scientific understanding, gathering knowledge first before locking experiments. Quality risk management in stage 1. Identify sources of bias and variability. Use process maps and Ishikawa diagrams. Rank risks qualitatively and quantitatively. Quality risk management helps identify and prioritize factors that impact method performance. DOE modeling and robustness. DOE explores interactions, model predicts performance, robust ranges are defined, designed experiments outperform one factor at a time, helping ensure consistent, reliable method performance. MODR and replication strategy. MODR defines safe and operating space. Replication improves precision. Focus on major sources of variability. The MODR shows where the method reliably meets the ATP, while smart replication controls overall variability. Analytical control strategy, system suitability tests, procedure, and environment controls, all designed to ensure the ATP is met. The ACS is the practical mechanism that keeps the method performing as intended throughout its life cycle. Stage 2 Performance Qualification. It confirms the method meets the ATP. It includes validation, verification, and method transfer. Protocol driven, fully documented. This stage aligns the traditional validation but within a life cycle framework. Stage 3 Ongoing performance verification. Routine monitoring, control charts, and trending. Early detection of drift and issues. Ongoing verification uses routine data to keep the method in control and fit for the purpose. Proactive, not reactive. Changes are assessed based on risk. The life cycle approach protects data integrity. Better science leads to better decisions. By closing the loop back to the ATP, analytical procedures evolve responsibility, stay reliable, and support confident quality decisions. This is the intent of USP 1220: stronger science, regulatory trust, and patient safety. Thank you for watching. Please subscribe us for getting update on upcoming industry application oriented videos. Please share your queries in comment section.